Okay, so next up we have Chris Knupp. He began volunteering in the Michigan State University Aquatic Animal Health Laboratory in 2015 while finishing his bachelor's degree in zoology. After that, he decided to pursue a master's degree in fisheries and wildlife with the Aquatic Animal Health Lab, where he researched the genetic diversity, virulence, and epidemiology of Flavobacterium psychrophyllum in North America. His master's degree left them with many new unanswered questions related to the disease ecology of Flavo, and so he decided to continue on as a fisheries and wildlife PhD student, where he is currently finishing his second year. So take it away, Chris.
All right, thanks for the introduction, Dr. Phelps, and I would also like to thank the organizers for putting this seminar series together. It's been really awesome watching everyone's talk. So I'd also just like to take a second to thank my co-authors for contributing to this project that I am excited to be sharing with everyone today. Okay, so as you all know, aquaculture now produces more animal-based protein for consumption than capture fisheries and is continuing to grow. This pie chart shows you that in 2018, U.S. aquaculture facilities raised nearly 80 million trout for market. What's important to take away from this figure, though, is that a third of these trout were lost to disease, which is a pretty substantial number. And what this figure doesn't show you is that hatcheries raising trout and salmon for conservation purposes are also losing their fish to disease. And so the losses are even greater than what we see here. According to the Food and Agriculture Organization of the United Nations, flavobacterial diseases are among the top 5 contributors to these losses. And as you can see in this photo, some flavobacteria are particularly damaging to their fish host to the point where they're essentially digesting the fish alive. So throughout this talk, I'm going to be using the term Flavobacteria to collectively refer to any member of the family Flavobacteriaceae. Now, when we're talking about flavobacteria that cause disease in salmon and trout, Flavobacterium psychrophyllum, which causes bacterial cold water disease, is believed to be the top contributor to disease-related losses in cold water fish species. But really, it's not that simple, because what we've been finding here in North America and elsewhere in the world is that there are a lot of emerging flavobacteria that cause disease signs that are very reminiscent of bacterial cold water disease. For instance, when you look at these images, you see that there are some fish that have disease signs that could be present in a cold water disease outbreak, but in fact, all of these fish were infected by different non-psychrophyllum flavobacteria. So we have psychrophyllum causing cold water disease, and we have novel flavobacteria that are causing disease that can be difficult to distinguish from cold water disease. So if that didn't make things complicated enough, well, in some cases, we have disease outbreaks where more than one flavobacterial species is co-occurring in the same diseased host. We call this a polymicrobial flavobacteria infection, where you have the presence of at least 2 flavobacterial species occurring in the same host. And so I just like to say that this isn't a rarity. When investigating flavobacterial disease outbreaks, it's not uncommon to recover multiple flavobacteria in the same host. So the overarching question that was at the core of this study was, how do polymicrobial infections impact the outcomes of bacterial cold water disease epizootics? These different flavobacterial taxa may have multiple biological differences, which could translate into differences in susceptibility to antimicrobial treatments or disease course or reservoir, or how they infect and enter their host. And so these mixed infections could have serious implications for how we may prevent, manage, and treat these disease outbreaks. But to answer this question, you really need an experimental challenge model to evaluate these infections under a controlled setting. So to begin answering this question, we designed an experiment with the following hypothesis in mind, which was that rainbow trout co-challenged with 2 flavobacterial taxa will experience greater mortality than rainbow trout challenged with each respective flavobacterial taxon alone. So the first thing we had to do in this study was we needed to decide what isolates we wanted to use. So the 2 most problematic genera in the family Flavobacteriaceae for fish, at least, are Flavobacterium and Chryseobacterium. Now, there are many different species to choose from, as you can see in these dendrograms, but we decided it would be best to pick genetically diverse isolates that look distinct from all other described species and were recovered from systemically naturally infected fish. So the initial part of this study, we chose 3 Flavobacterium isolates and 2 Chryseobacterium isolates, and these fell into different clades. So as a starting point, we then wanted to evaluate the virulence of these 5 Flavobacteria under controlled conditions. So to do this, I had intramuscularly injected 10 to the 7 colony-forming units of each Flavobacteria in a 1.5 gram rainbow trout. This figure shows you that the cumulative percent mortality caused by those 5 Flavobacteria. So as you'll notice, we didn't have any mortalities in our mock challenge fish. Next, I would like to highlight that not all of these Flavobacteria were pathogenic. So as you can see here, fish that received 10 to the 7 CFUs of C3, none of those fish died. However, the other 4 Flavobacteria caused varying amounts of mortality. So at this point, we wanted to pick the Flavobacterium species and the Chryseobacterium species that had caused the most mortality. And so that was isolates S21 and T28. So next, we estimated the median lethal dose of these 2 isolates to guide further challenge experiments. So to estimate the median lethal dose, we wanted to start with a reproducible challenge route for these novel Flavobacteria. And so we decided to use intramuscular injection. So we intramuscularly injected a range of doses into 2.5 gram rainbow trout. And this figure is showing us the cumulative percent mortality at each of those doses for Flavobacterium species S21. Using this information, we estimated the median lethal dose for S21 to be 5.48 times 10 to the 6 colony forming units. And so just so you guys have a frame of reference, the median lethal doses obtained for Flavobacterium psychrophyllum, which fall under the range of 10 to the 2 to 10 to the 6 CFUs, as has been reported in several different studies, including those done early on by Holt. So here are the challenge results for Chryseobacterium species T28, which again was intramuscularly injected into juvenile rainbow trout at a range of doses. And so the median lethal dose for T28 was 1.58 times 10 to the 7 CFUs. All right, so now we had our novel Flavobacteria, but we needed to select a Flavobacterium psychrophyllum isolate to co-challenge our fish with. So here I'm showing you the global population structure of Flavobacterium psychrophyllum according to multi-locus sequence typing. Now, I'm not going to get into all the details of this other than to say that we use this data to select sequence type 78, which has been recovered from multiple disease outbreaks across the United States. And based on our previous experimental challenges using isolates from this genotype, we know that they are virulent. So now I'm going to take you through the polymicrobial experiment. So the first step was to culture these Flavobacteria for the experimental challenge. So the psychrophyllum isolate was cultured in a modified Ties broth, shaken for 48 hours at 15 degrees Celsius. And then I harvested the cells and quantified them using plate counts. The 2 non-psychrophyllum Flavobacteria were cultured in a modified stew broth, which was grown statically for 48 hours at 22 degrees Celsius. And then I harvested and quantified those as well. Now, before getting into the specific treatment groups, I wanted to take you through how I challenged these fish. So here we have our rainbow trout. And the first thing I did was anesthetize it using MS-222. And then I removed the adipose fin. Next, I intramuscularly injected one of 3 options, either Flavobacterium species S21, Chryseobacterium species T28, or saline. So after this, fish were then immersed in one of 2 options, either just water or water-containing psychrophyllum. So as I said before, we decided to use intramuscular injection for the non-psychrophyllum isolates, but given that we wanted to see how these different Flavobacteria were interacting with psychrophyllum, we really did feel that it was important to challenge the fish via immersion with cycrophyllum because it is a more natural exposure route. Now I just want to briefly go over the individual treatment groups. So like I said in the previous slide, fish were first anesthetized and then injected with, for example, 50 microliters of T28 at a high dose. And then these fish were either immersed in water only. So that's going to be your T28 high dose control group, or a high concentration of cycrophyllum, or a lower concentration of cycrophyllum. So each treatment consisted of 30 fish, which could be broken down into 15 fish per replicate tank. So we repeated the process, but fish were first injected with T28 at a lower dose, and then immersed in either water, a high concentration of cycrophyllum, or a lower concentration of cycrophyllum. And so the same process was repeated for S21 and for the other control groups. So now I'm going to go through the results of what each bacterium did on their own before showing you how they did together. So this figure shows the cumulative percent mortality caused by Flavobacterium cycrophyllum alone at each of the 2 doses over time. The different treatments are denoted with different symbols. One thing I'd like to say right off the bat is that most cycrophyllum challenge studies only last for about 28 days. But so we chose to carry out the experiment for 14 days after the last mortality, because some of these polymicrobial infections, they may be a little bit more chronic in nature. So in this figure, you can see that the cumulative mortality for fish immersed in a high concentration of cycrophyllum was about 10%, whereas the mortality for fish immersed in the low concentration of cycrophyllum was 0%. Here we're seeing a similar figure, but with Chryseobacterium species T28 only. And so you can see at the high dose, it caused about 57% mortality, and at the low dose, 0%. Okay, so this figure incorporates the results of the single infection treatment groups with the co-challenge treatment groups. So those co-challenge treatment groups, you can see that those are represented by the red symbols. So drawing your attention to this line here, we can see that this treatment group that was given a high dose of each Flavobacteria experienced 73% mortality. And so if you add the mortality that each of these isolates caused when they were alone together, you get 67%. So that's not too different from 73%, suggesting to us that maybe this is an additive effect in the co-challenge group. And now when you're looking at the group given a high dose of T28 and a low dose of cycrophyllum, we see that this co-challenge group experienced 63% mortality. If again, you add the mortality from each of those individual isolates together, when they were co-challenged alone, you get 57% mortality, which is fairly similar to 63. So at this point, it appears that mortality seems to be merely additive. So this is Flavobacterium species S21 only. And so you can see that there was about 27% mortality while we were at the low, or while we were at the high dose, and at the low dose, we're looking at about 0% mortality. So here are the results again, this time with Flavobacterium S21 and cycrophyllum. So if I draw your attention to this line here, what we see is that the treatment group that was given a high dose of each Flavobacteria resulted in 47% mortality. And if you add the mortality from each of those isolates when they were alone, you get 37%, which is slightly lower than the co-challenge group, about 10, it's 10%. Now when we look at the treatment group that's given a high dose of S21 and a low dose of cycrophyllum, we see 57% mortality overall, which is 2 times higher than if you add the mortality from each of those individual isolates alone. So at least based on this data, it seems like we're getting something here that's more than additive. is it an indication of some synergistic virulence, or, are these Flavobacteria working together against the host? At least based on this data, we feel that this could be the case. Now, no doubt you noticed, but we got this unexpected outcome where the mortality in this co-challenge group exceeded the co-challenge group that actually got both bacteria at the high dose. These two. And so, I guess we really shouldn't have been too surprised, since we're working with multiple novel Flavobacteria, that we were throwing a bit of a curveball on this one. Okay, so I just showed you the mortality. How about the infection status of the fish that died during the experiment? Well, in the single infection groups, I was always able to recover the bacterium that was used for the challenge. However, the results for the co-challenge groups were a little bit less clear, in that we always recovered the non-Psychrophyllum flavos, but because these non-Psychrophyllum flavos could also grow in the same culture medium as Psychrophyllum, we couldn't always recover Psychrophyllum, and that was likely, we think, because it was being outcompeted. However, there were some instances where we could clearly see Psychrophyllum growing underneath the non-Psychrophyllum flavos, and so future studies could definitely improve on our methods by using, a molecular technique, qPCR or something like that. So this is where things got, even more interesting for us. So just to orient you, we have the treatment groups on the x-axis, and each has 2 bars, one for each replicate. And on the y-axis is the number of fish that survived 65 days post-infection. Now I've separated the single treatment groups from the co-challenge groups using that yellow line. So this figure is showing you how many fish either had Psychrophyllum, which are the black bars, or S21, which are the red bars, in their kidney 65 days post-infection. So you'll notice neither Flavobacteria was recovered from the negative control fish. You can also see that we never recovered Psychrophyllum from the kidney, and either in the Psychrophyllum only treatment groups. In either of those. And we never recovered Psychrophyllum from either the S21 low or the S21 high treatment groups. But you can see that when we start looking at these co-challenge treatment groups, that we are recovering at least one fish from each of those co-challenge groups has one fish with Psychrophyllum. And so this figure is showing us the same thing as the previous one, but with Chryseobacterium species T28. So again, you'll notice that we didn't recover either Flavobacteria from the negative control fish. We did not recover Psychrophyllum from the Psychrophyllum only fish or from the T28 only fish. But you will see that we did recover Psychrophyllum from the kidney of at least one fish in all but one of the co-challenge groups, at the end of the 65-day experiment. So to us, this was, really unexpected and interesting, because the way that we interpret this is that when you have fish that are infected with multiple Flavobacteria, that this, appears to prolong the infection status for Psychrophyllum, which, could have implications for reservoir status and the perpetuation of infection. And so, this is something that, not only are we seeing here with T28, but we saw it in the previous slide with S21 as well. And so I also wanted to note that with T28, which again is a novel species that we know very little about, that this bacterium persisted, in the face of the host immune system for 65 days, in the kidney at a relatively high prevalence. All right, so now that I've taken you through the effect the effect on cumulative percent mortality and the effect on long-term infection status, now I'd like to take you through some of the gross manifestations of disease. So just for reference, this is how the adipose fin clip site appears on the negative control fish following the removal of the adipose fin. So here we can see that fish that received the non-Psychrophyllum isolates only had ulcerations that are reminiscent of those present in cold water disease outbreaks. Now, comparatively, fish that received cycrophyllum alone had the same lesion, albeit more severe. So when we look at some of the co-challenge groups that receive both isolates, we see that the lesions are even more severe. And now what's interesting is that in these co-challenge groups, we are seeing severe, these two bottom ones, we're seeing severe lesions, even though that neither of these Flavobacteria alone could cause these lesions. So when you pair this with what we saw with the cumulative percent mortality for S21, we feel that this is further evidence that these Flavobacterial taxa may be synergizing and working with each other to elicit a more severe disease response. So I just showed you what happened on an external site. So what about systemically? Well, here you can see that some of the fish that were challenged with cycrophyllum only exhibited exophthalmia. And in the other treatment groups that were challenged with only one Flavobacteria, we didn't see any exophthalmia in any of those fish. So when we look at some of the co-challenge groups, we also are seeing fish with exophthalmia. But again, what was interesting to us was that with these groups, none of those isolates that make up those co-challenge groups were causing exophthalmia on their own. However, when you combine these Flavobacteria taxa together, we're starting to see exophthalmia. And so similar to the last slide, it does appear, at least in this experiment, that there's potentially a synergism between some Flavobacteria, when they're infecting the same fish. So in conclusion, I think that we were able to design a workable challenge model for polymicrobial Flavobacterial challenges. We did have some variability and unexpected results, but that doesn't surprise me too much, especially considering that we're working with difficult microbes and, not only that, but trying to manage them at the same time and the same host. And next, so sometimes it appeared that, some of the treatment effects in the co-challenge groups were additive, which was largely the case for cycrophyllum and T28. But in other cases with cycrophyllum and S21, we were getting substantially more mortality in the co-challenge groups. And like I said on the last couple of slides, when you pair that with what we were seeing with the external pathology with the lesions at the clip side and the exophthalmia, it seems like there is something more than just additive going on. So I think this could make a pretty good case for having some synergistic effects, and maybe that depends on the Flavobacterial taxa that are co-infecting the same fish. And so something else that we didn't expect was the long-term infection status for fish, that were challenged with multiple Flavobacteria, harbored cycrophyllum in their kidney for a much longer period of time. Like I said before, this could have implications for, reservoir status and subsequent infections, later on down the road. And so with that, I'd like to thank the USDA for funding this project and Michigan Department of Natural Resources, and of course my advisor, Dr. Tom Locke, and the VDL and everyone that's back at the lab. So with that, ready to take questions.
Great. Thank you very much, Chris. Excellent presentation. We've got a few minutes for questions, so go ahead and unmute yourself or put it into the chat box.
Hey, Chris, this is Jan. That's a cool presentation. Nice experiment that you did. I was just curious, maybe I missed it. How much time went by between the initial injection of the Flavobacterium before they were immersed with cycrophyllum?
Oh, yeah, I guess I didn't quite go over that, but it was immediate.
Okay.
So the fish would be clipped, injected, when they would be allowed to recover first before going into the the bath with cycrophyllum in it. But so yeah.
So do you think that you're still giving a little bit of a head start to that initial Flavobacterium by injecting it first and then putting cycrophyllum in there. And to get to that, I wonder if you would design an experiment where you immerse them with both bacteria at the same time, and maybe you see different effects. Maybe the less pathogenic Flavobacterium can outcompete the more pathogenic cycrophyllum for binding sites and stuff like that. Is there any intent to try to address that, or do you think that that's a possibility?
Yeah, that's actually a really good point because we had chose to inject the non-cycrophyllum flavos, and I actually didn't cover it in here, but so we had actually tried several different methodologies for using an immersion route with those flavos, but we weren't having too much luck with them, and you know, I'm not really surprised by that very much because people are still having, you know, difficulties using cycrophyllum for immersion studies, even though, you know, we've been studying it for 80 years now. You know, I do think, you know, if we, you know, future studies, it would be good now to maybe, do what you're saying and immerse the two together and see, you know, what they can do. But, you know, at least at this point, it seems, you know, if you can get that non-cycrophyllum flavo into the fish first, and then, you know, cycrophyllum comes along and is able to, you know, manifest, you know, this is what we get. But yeah, I think that would probably be a, you know, a good next step.
Cool.
Challenging them together through immersion.
Yeah. All right. Thanks, Chris.
Yeah.
Hello. Hi, nice talk, Chris. So this is Colin from the UPI. I have like a general question regarding to the challenge model, and also this maybe also question to Tom or Ben. Like, which one do you recommend? Like for the cycrophyllum challenge, is IM, IP, or immersion? I ask this question because we also do some this kind of challenge in our lab, but I got like I did several tries. It's got a 0 mortality. I noticed your experiment also like the mortality will be like 20%. It's kind of low. Like, and also your fish is kind of like I want to double check it's like 2.5 gram. So, but we use like 50, 60 gram fish. I wonder like which kind of like challenge model you guys recommend, and you know, how to you know, to improve, or what is the more consistent challenge model?
Mm-hmm. Yeah, so I think you're gonna get maybe some different answers, but when you go and look in the literature, at least right now, most people are saying using intramuscular injection for cycrophyllum is going to produce the most reproducible results. And that's going to be kind of independent of the size of the fish. You know, we wanted to use immersion because it's more natural, and people have tried to use immersion, and they do use immersion, but it can be difficult, you know, depending on the isolate. So it's not always so clear cut, you know, it's like not really like a one size fits all with cycrophyllum and immersion. You know, some isolates might need different things, but generally it seems like if you're better off using maybe smaller fish, or at least that's what people report in our experience. Using smaller fish for immersion seems to help. But yeah, I guess it kind of depends on what you're after, because I would say most people, when they're doing virulence studies on cycrophyllum, they usually tend to go towards IM just because of how much more reproducible it is. And it gets the, you know, cycrophyllum, you know, straight into the fish, but of course it, you know, avoids the more natural part of cycrophyllum having to gain entry. But we have been able to do studies with cycrophyllum via immersion and working with LD50s. So it is possible. It just takes a lot more fine tuning, I guess, is what I would say.
Okay, yeah, thanks. I think my next try probably should be like a try intramuscular.
Oh, yep.
And also at the immersion, which didn't work well.
And we've got time for one more quick question.
Hey, Chris, this is John from Colorado. Hey, great talk, and actually really complicated stuff. Actually, it's really neat. Can you kind of give insight on the two things? why do you think you had such a drastic, when you're doing your challenge model doses? I mean, you went from like 10 to the 7 and then nothing ever, from 10 to the 6, 5, 4, 3, it just dropped down to almost nothing. Like you didn't see a dose dependent response, or you didn't see a dilution effect, and like your mortality was high and then it went down to basically nothing. And then the other question is, when did you guys sample the kidney tissue for FP or Chryseo? And I've seen in a number of times where I can't explain it, but Cycrophyllum tends to have an affinity for spleen tissue, and we find it more often in early infections in the spleen, but not the kidney. And conversely, you can find other bacteria only in the kidney, but not in the spleen. And I'll just get thoughts on that one too.
Yeah. So yeah, with the IM and the median lethal dose for those non-Cycrophyllum flavos and the big jumps, you know, I guess my thought is, you know, we didn't have, I mean, the variation between the replicates was, you know, pretty much non-existent. So I don't really think it was an effect on, you know, injecting them in different ways. I guess, you know, what I think when stuff like that happens is, you know, maybe some of these doses are, you know, maybe it needs, you know, people like to go like 10 to the 7, 10 to the 6, but maybe it needs to be a little bit more fine tuned. And maybe that could even be like a better next step sometimes. You know, you figure out where it falls generally with those, and then you go into it a little bit further, which can be difficult to do, you know, trying to get reproducible doses and everything like that. And then with the kidney, so I was culturing from the kidneys throughout the experiment, but the histogram that I showed, so that was kidney cultures 65 days out at the end of the experiment. Yeah, and then I'm sorry, I kind of forgot what the last part about the kidney and the spleen.
I actually, the kidney 65 days out as a reservoir. That's interesting.
Yeah.
It's just that we tend to see Cycrophyllum in the spleen first often compared to the kidney. Similarly, Flavobacterium succinicans, we only find that in the kidney once it gets in, but it's not in the spleen. Go through a number of examples like that.
Yeah, I mean,
just curious at spleen and seeing the bacteria, if the bacteria is still there, but in the spleen and not the kidney.
Mm-hmm. Yeah, I mean, we pretty reliably recover Cycrophyllum oftentimes from the kidney, and you know, it's a excretory organ, so you know, we like to target that one. We do. I mean, I've done other challenges with Cycrophyllum, and you can recover, you know, similar loads of bacteria from the spleen too. And I did save a bunch of the spleens from these fish. So, you know, maybe if I feel like I want to do some more work on this or something, I can go back and, you know, do some qPCR on the spleen tissues and see what's going on there too. But yeah.
Regardless, that was a great talk. Thanks.
Yeah, thank you very much, Chris, and everybody for the discussion. That's our time for today. So we've got 2 more interesting presentations next week, so please come back and invite your friends for the second to last day of the seminar series this summer. So everybody have a great week, and we'll see you then.
Thanks, Nick. Thanks, everybody.